Hi everyone. Uh, okay. Hi. It's the first time to do the webinar in Malaysia. Usually we will go to Malaysia. We will discuss some topics face to face. But in this case, we have COVID-19 pandemic severity. So I think it's the only way to communicate with you through the internet. I see here nearly 100 people here online, so let's start our speech here. So I will shut down the video and share with my screen. Okay, TransGen biotech we have found for 15 years, and we focus on the molecular and the cell biology experiment. Today's topic is about the qPCR, qRT-PCR systems. Next week, we will focus on the cell culture system. So this one is based on the molecular biology, and next week, we will focus on the cell biology. Now, today's speech, there are five parts about today's speech. The first one is RNA extraction. So we have to extract RNA from the samples, from the materials. And then we will do the reverse transcriptions to make the RNA into cDNAs. And then we will do the qPCR to detect the specific genes expression levels. And then we do the data analysis, and we will find out whether or not this is a good result or not. And the last part is amplifications. So here we focus on the first one, RNA extraction. High quality of an RNA template is the basis of the successful follow-up experiment. We have to pay attention about the RNA's qualities. We have to focus on the preservation of the materials and also the RNA extraction and the quality controls. And also GDNA contamination is also a very important factor during the RNA extractions. First of all is the preservation of the materials. The fresh materials should be immediately extracted or frozen by liquid nitrogen. Because during the whole experiment, the RNAs will be released from the lysosomes to degrade the RNAs. So with the liquid nitrogen, the RNAs will be frozen in very quick steps. So in this case, liquid nitrogen will protect the RNA into a very good circumstances. But during the whole experiment, always there's no liquid nitrogen exists. So in this case, we can use RNA hold from our complex to provide another protection for your RNAs. RNA hold, the main components of the RNA hold are non-specific protein denaturants. It can rapidly permeate tissues and cells, denature all the proteins, such as RNAs, and protect the RNAs. This experiment will show you the result about the RNA hold. We used human total RNA from the HeLa cells. As we can see here, the fresh material is extracted immediately. We showed very good result about the RNA. We showed the very specific band of the RNAs. And this one, we used RNA hold and preserved the materials at the 37 centigrade 1 day. We can also extract the RNA very well. But without the RNA hold, the whole RNA degrades from the systems. And when we use the RNA hold, we can see the longest day we can hold the RNA for 4 weeks during the experiment. There's three main methods for RNA extraction. First one is the grenadine method. The grenadine can lysis the cells and block the RNA's activities, retain the RNA completely. And also the others, chloroform or silicon membrane can be used for RNA extractions. This kit is based on the grenadine method. Also, the other one is the CTAB method. CTAB method will also lyse the cells and release RNA, and it will avoid polysaccharide and polyphenols binding the RNA. So it's very useful for plant materials, so such as transgenic plant is based on the CTAB method. And also the next one is the magnetic beads method. We used a unique lysis buffer to lyse the virus and release the DNA or RNA. And release the DNA and RNA will be effectively purified and specifically binding the silicon magnetic beads. So when we use the Magic Q, this brand, we use the magnetic beads method. When we get the RNA, the quality control of RNA is very important. We have three different ways to do the quality control. The first one is agarose gels electrophoresis. The other one we use the machine we call the nanodrop machines. And the other one we use the Agilent RNA lab chip and the 2100 biolyzer. These machines can be used to detect the RNA's completion. First one is the agarose gel electrophoresis with very high voltage in a very short time for the electrophoresis, maybe 5 minutes at 200 voltage. We can see the very good result about the RNAs. There's a three band here. The first one is the 28S RNA, the other is the 18S RNA. And these two bands, the ratio between these two bands is 2 to 1. And also it's very slice here is 5.8S RNAs. But in this case, the RNA is not so good. So this is the degraded RNA. And so it's very smear band here. There's no specific three bands. And also 5.8S RNA is shows a very large amount about that. It shows the RNA is totally degraded. And also this one, this band, we call it the genomic DNA. Genomic DNA during the RNA extraction is a very bad solution, and it will cause wrong data for the qPCR systems. The other one is the nanodrop detection. With this machine, we just need maybe 1 or 2 microliter to detect RNA, and we will easily get the concentration of the RNAs. And the other two, the key points about the system, we can get the ratio between 216 to 280. And the pure RNA about this ratio is 2.0. And the pure DNA, usually is 1.8. And the protein contamination significantly reduce this ratio. So when you this ratio is maybe 1.0 or 0.7, maybe there's a lot of protein contaminations. And the other one is a ratio between 260 to 230. And usually this ratio is 220 to 2.2. And also the light-absorbing substance residues at the 230 will be significantly reduced at the ratio, such as EDTA, phenols, guanidines. So maybe the such as organic solvents will reduce these ratios. All these ratios means there's impurities about the RNA. So maybe these impurities will become the inhibitors during the RT system or PCR systems. The third one is we could Agilent bioanalyzer. With the bioanalyzer, we have to use also the RNA chip. RNA chip is very expensive. Maybe during the whole experiment, when we detect one sample of the RNA, maybe it will cost $7 to $10 to detect one samples. So it's very expensive. But when we use the Agilent 2100 bioanalyzer, it will get a very precise result about the RNA's qualities. So here is the very good quality about the RNA. We can see the three individual peak here. The highest one is the 20S RNA, and the other is the 18S RNA and the 5.8 RNA. So we can see the very individual peak, just like the electrophysics, the three bands here. But in this case, the degradation RNA shows very chaos signals here. Shows the RNA, the quality is very bad. So when we use these machines, it's very expensive. So if we have to focus on the integrity about the RNAs, we have to use this one. Usually, when we do the next generation sequencing, we will do the RNase lab prep. We have to use these Angelins 2100 systems. But when we do the qPCR system, maybe this one is too expensive for the qPCR. We can just use the nanodrop detection and the agarose gel electrophysics. We can do the very good quality controls about the RNAs. Okay, the next one is genomic DNA contaminations. This contamination will influence the result of the qPCR. In the systems, the genomic DNA will also can do the qPCR systems. So it will influence the whole system and overestimate the result. When we use the good quality reagent, we can avoid gDNA contaminations. Also, setting controls, we can detect the gDNA contamination to avoid influence. We have discussed a very important part about the RNAs, but when we dealing with the RT system, we have to face on the very three key factors about the RT efficiency. So it's the RNA, primers, and artemies. So earlier we have to discuss ago. So we have to focus on the primers and the artemies. There's three different primers for the RT systems. The first one is the random primer. Random primer is just like a random N6 or N9 nucleotides. So it means it will randomly bind to mRNA. So it will show very high efficiency for the RT systems. But also it will maybe overestimate the result because the random primer not only just binds the mRNA, it also can bind different kind of RNAs such as RNA, tRNA, or surface RNAs. So it shows a little risk for the overestimated result. The other one is the oligo-DT primers. Basically, it's just oligo-T, so TTTT. These Ts can specifically bind to the poly-A tails of the mRNAs. So oligo-DT always is a specific primers for the mRNA synthesis. And the third one is the sequence-specific primers. These primers are not bind with the mRNA's structures, just bind with the specific genes sequencing. So when we use the sequence-specific primers, it's usually for the one-stop qRT-PCRs. So we will combine with the qPCR, RT and qPCR together to share the same primers as a sequence-specific primers. When we use the oligo-DT as a very specific mRNA systems, so it's very efficiency and it's very good. It's very widely used, but it also have this disadvantage. During the whole systems here, we can see when we primers bind with the poly-A at the 3 ends of the mRNAs, so the cDNA synthesis, the direction of the cDNA synthesis is from 3 end to 5 end. So when there's a complex structure during the mRNA, the whole synthesis system will block. So it means the information in this area will be lost. So when we use oligo-DT, maybe there's a risk to lose a lot of information away from the 3 ends. But in this part, we put the oligo-DT and the random primer together. When the oligo-DT do his job, and there's a complex structure block the oligo-DT synthesis, and the random primer can cover the rest of the sequence. So it will get more information about the whole systems. But in this case, the complex structures information also be lost. So in this case, we have another solution for the systems. Artemis, artificial activity can do the synthesis based on the DNA or RNAs, but the activity based on the RNA is much higher than the based on the DNAs. So the main activity about the polymerase is the Artemis systems. And the other way, there's two different kind of activities we have to focus on. First one is RNase-H activity. The other is the thermal stabilities. The RMH activity shows it will degrade RNA, which just bind with the CDNAs. The individual RNA will not be recognized by RNase-H. So in this case, we have to delete the RNA-H's activities. Unfortunately, the RNase activity domain is separate from the polymerase domain. So it means we can do the site mutation or domain deletion to release the RNase-H activities. So in this case, it will increase the length of the CDNAs. The other is the thermal stabilities. This image we just discussed that later. When we do the incubation at the 42 centigrade, it means a lot of information away from the 3 end will be lost. But if we put the system into very high temperature, such as 50 centigrade or even higher than to 65 centigrade, it will open the complex structure of the RNAs. It means the information during the complex structures will also be released and also be detected by the whole systems. In this case, it means Artemis should have the very higher thermal stabilities. It can survive during the 65 centigrade or 50 centigrade. So in this case, thermal stability also is a very important factor we will deal with our reagent. So here we have 5 different core enzymes about the reverse transcriptase. The easiest script Artemis is 101, so it's very basic one. The other is the transcript Artemis. It just do the site mutation to increase the synthesis and the efficiency. And the transcript 2 Artemis and the transcript uni Artemis do the site mutation to increase the thermal stability. Transcript 2 Artemis can survive at the temperature of 55 centigrade, and the transcript uni Artemis can survive at the temperature of 65 centigrade. So it means if you have a very very complex structure of RNA or high GC content, maybe up to 60% of the high GC, it means you can use this transcript uni or transcript 2 Artemis to do the RT systems. And the other one is the transcript fly Artemis. It only can survive at the temperature of 42, so it's not shows very good quality about the thermal stability. But it do the segmentation to increase the process. It can do the whole experiment very quickly, just in 5 minutes to finish the whole systems. So when we do the fly, it means it's so quickly to do the experiment. Here's a result. Here's a question here. In experiment, is it possible to isolate high quality RNA from the formalin-fixed human tissues? If the quality is not good, can we still get the reliable qPCR results? Yes, it can. But we have very focus on this formalin-fixed and This formalin fixed tissues will destroy the proteins and the nucleus. So it means the DNA and the RNA also be destroyed. So when we do the extractions, we can see the DNA and the RNA is a very smear band, not the specific 3 band of the RNA. So in this case, RNA also be destroyed. But also we can do the result, and this is a reliable result about the qPCR systems. We have to accept that the RNA is not good, but the result can be get. Also, in this case, when we do the formalin fixed human tissues, the RNA from that also can do the next generation sequencing. We also can get the reliable qPCR result, because everyone knows that in this case, the formalin will destroy the systems. Okay. So if you have any questions, so you can leave here, and our colleagues will collect all the questions, and we will answer the questions at the end of the speech. Okay. So nowadays we have a very new product here. Excuse me. Okay, I release that.
哦，
那你休息点。昨天怎么去的你昨天就是这么拿的。Okay, let's.
就到头了。
Let's in. Okay, it's this. Okay, nowadays we have a very new products. Is combined with this fly Artemis and the uni Artemis. So this very new one, it can survive at the temperature of 65 centigrade, and also it can react just 5 minutes. So it's a very new product here. So we can choose different kind of products based on the sensitivity, fidelity, GC-rich complex templates, and so when we get the RNA and we do the RT systems, we get the qPCR, the cDNA. So it means we finally can do the qPCR experiment. During the qPCR, we have to focus on the normal one, the PCR system. When we do the PCR system, it will detect the end products. The end products of the applications reaction were analyzed semi-quantitatively and qualitatively by electrophysics, such as like this one. But in this case, it shows very smear results, so it means it's not very accurate. So during this nowadays, we have to focus on the qPCR systems. When we do the qPCR, the change of the products in each cycle of the PCR application reactions was detected by the change of the fluorescence signals, and the initially template was finally analyzed and quantitatively. So these are two different here. Is PCR detect the end of the products, and the qPCR detect the initial templates. So it's a totally difference between this PCR and the qPCR. When we do the qPCR system, there's several curves is very important. The first one is the amplification curve. This is the raw data of the amplification curve. It shows here. The baseline is a very chaos signal shows here. So this is the raw data. When we see this raw data, we cannot detect any useful signals, information from this chaos. So the machines will do the job. They will do the standard lines here. So shows very smoothly line as a application curves. And also there's a threshold line. The threshold line corresponds with the application curves, and this one is the CT value. So this CT value is very key factors during the later calculation systems. And the other curve is melting curve. Now when we do the melting curve, it detects the specificity about the whole experiment. The most ideal is a single peak. The peak is very narrow. If there's multiple peaks, so it shows the reaction is not specific, or there is a primary dimer exists. During the qPCR, there's a three methods for the qPCR system. First one is the subgreen-1 method. Subgreen-1 is fluorescent dye. It will not release the signals just until he binds with the double-stranded DNAs. So during the PCR systems, a lot of more and more products shows the double-stranded DNAs. So the subgreen-1 will bind with this gap between the DNAs and shows the fluorescence. So in this case, it's very cheap. It's very easy to operate. So this is just a PCR with dye. So it's very easy one, and it's very cheap reagent. The other one is the Techman probe method. With Techman probe, without the subgreen-1, just with the Techman probe is that. So in this case, the Techman probe is a very small probe to detect the specific target genes here. And it has two factors here. One is the reporter dye. Reporter dye shows the different fluorescent signals. The other is quencher. This quencher can banish the reported signals. So when the Techman probe is complete, there's no signals released. And then when we do the PCR systems, the polymerase will cut the probe and then release the reported dyes. So the signals will be detected by the machines. In this case, we use different probe with different fluorescence signals. It means we can do the multiple PCR during the whole systems. And the specificity about the experiment is much higher than the subgreen-1 systems. The third one is the hybridized probe method. Is we call the molecular beacon. Molecular beacon is very long. So in this case, also the fluorescence dye and the quencher will bind together, shows nothing signals will release. But when the target sequencing exists, this probe will be hybridized with the target genes and then release the fluorescent signals will be detected by the machines. So in this case, the probe is very long, so it means it detects very specifically. So it can use for SMP system. One step versus two-step qPCR. One step means in one tube with the gene-specific primers, you can just get the RNA into the applications in one tube. So it's very easy to operate, but it just can be detect specific gene primers, such as chromatins detection, we use the one-step RT-PCR system. And the other is the two-step RT-PCR with two tubes. The first one, we use the oligo-DT or random primer to get the cDNA, and then get the cDNA with the help of the specific gene of the primers of the targeted genes. We can get the amplifications. So this is two-step. In this case, it's very suitable for small, very fewer RNA, and we have to detect a lot of multiple genes. We can use for we can use the two-step RT-PCR system. In internal reference gene selection, during the persistence, the internal reference gene is a very important one. With the help of the internal reference genes, it can detect the degradation of the RNA in the sample materials, and also it can correct the sampling error and the cDNA synthesis rates, and also it can correct the effect of the PCR inhibitors during the whole systems. And the common reference genes, we can use the GAPDH, beta-actin, 18S RNA. This three is the main common reference genes during the whole qPCR systems. And also, when we do the qPCR, we have to design the primers. There's several principles here to when you design the submarine primers. First one is very important one. The length of the primers is 18 to 30 pp, and the range of the products is 100 to 400 pp. And also the GC content is at the range of the 40 to 60%. And also it should avoid primers containing the complementary sequencing, and also avoid 3-end mismatch, and no more than 3 consecutive G and C, and avoid the T at the 3 end. So after we prepared all the primers, cDNAs, the last one we have to design our qPCR experiment. When we do the biological experiment, the control always be first considered during the whole systems. There's two different kinds of control. First one is negative control, and the other is the positive control. When we do the positive control, we can use the plasmids, cDNAs, standard curves can also be the positive control. It just shows the system can work very well. And the other is the negative control. Negative control shows our systems worked very precisely, and the result can be reliable. So there's two things. First one is the NTC. NTC means no template control. It checks the template contaminations. And the other is NRC. NRC means no reverse transcription control, because of no RT control. It will test RNA sample for DNA contaminations. So this NTC and NRC are both very important negative control during the qPCR systems. Maybe you publish your data to the articles. Maybe these two controls do not to show to the editors, but it has to be detected in your experiment. Subgreen 1 method, it has a very easy experiment during the whole systems. So it means the copy number of the gene should be more than 1,000, because it shows less specificity about the experiment. So it means the copy gene should be very large amount. And the other is the pre-test before the probe qPCR, because the probe qPCR is much more expensive than the submarine one. So it can be used for a pre-test before the probe qPCR. And also the PCR condition should be very mature, no dimer, no non-specific applications during the systems. And the TechMan Pro can be used for multiple PCR. With the labeling different, the report groups can be used for multiple PCR. In our case, our detection diagnosis system for COVID-19, we did 3 multiple PCR and 4 multiple PCR different kits. And also the experiment with very high sensitivity is a requirement, and also can be used for diagnosis systems. ROX control signals. ROX is also a fluorescent signals, but it will not influence with anything in the systems. Subgreen 1 can react with the DNA, double-stranded DNA, but the ROPS will not influence anything. So in this case, with a standardized fluorescent signals, it will adjust errors caused by non-PCR factors, such as sampling errors, or it will provide very stable baselines. A lot of instruments, a lot of machines need the rocks as a standard fluorescent signals to calibrate his machines, such as ABI experiment. ABI machines always use ROX1 as a standard fluorescent signals, and also such as Bio-Rad, Roche, you will use any ROX control signals. Here we provide two different reagents for the qPCR supermix. First one is the perfect step green qPCR supermix. The other is the Pro. So it's very easy to separate them. Green means subgreen 1. Pro means the Techman Pro. So we can choose based on your experiment. When we finish our qPCR, we have to analyze the qPCR's data. Evaluate the validity of the data. There's 3 positive results and 2 negative controls. The 3 positive results here is the amplification curve, melting curve, and the standard curve. And the 2 negative controls is the NTC control and NRC control. First of all is the amplification curve. Amplification curve should be very smoothly, no such chaos signals, and also no amplification at the first 10 cycles. So there's no signals in the first 10 cycles here. And also the CT value are normally 15 to 30. This will be very good result here. And there's no application of the NTC and NRC. Usually the credible range of the CT value, when we do the clinical detection, always less than 33. And also science research is less than 38. And the reference gene always less than 20. And the duplications should less than 0.5 between the duplications. But we have to pay attention here. The clinical detection for diagnosis always less than 33. But there's one exception. Here, when we do the COVID-19 detection, because the COVID-19 is a virus, the RNA from the whole system is very rare. So in this case, when we do the diagnosis for the COVID-19, maybe the CT value is larger than 33. Always, it's less than 40 will be accepted. The melting curve. The melting curve should have just one single peak, and the range of the peak is narrow. It shows very good result and shows very specifically. Though there's no peaks appears in NTC and NRC, it's a very good result shows the melting curve. And the other one is the standard curve. When we use the standard curve with this formula here, we can calculate the application efficiency during the standard curve. When we put the standard curve, we will gradiently dilution the template, maybe the 10 times or 4 times, and then we do the qPCR. The CT value shows this arithmetic sequence about the whole systems. So it shows in the image the all dots combined with the one line here. And this line, the formula shows this R squared is nearly to 0.999. So this one, R factor, is called the regression coefficient. This regression coefficient square should be at 0.95 to 1. So this is very good range about the whole systems. So in this case, with this formula, we can calculate the application efficiency. So the efficiency should be at 19% to 111%. So when we design your own primers, you have to calculate the application efficiency. If your efficiency is lower than 90%, the result may be not reliable. But if you when you do the experiment and calculate the efficiency in this range, it shows your design primer is very good. And then is the negative control, NTC, no template control. It means the whole systems with any components in the systems just without the templates. So in this case, it will the good NTC will show nothing signals here. So the best data of NTC is not detectable in this one. But always in the systems, maybe there's little signals at the very the end point of the whole systems. So when we loosen the criteria, there's more than 25 CT value can be acceptable. Just like this one. This is a very good result of the NTC. There's another detectable NTC. But this one, NTC, it shows little signals here. But the TM temperature is lower than 80 centigrade. In this case, we will focus on this. We will recognize this as a primer dimer. So in this case, we can accept this result as a reliable result. The next one is no RT control. It means whole systems without RT system. So it means in the whole systems, the template just RNA with no CDNAs. So it's a good result for NTC. Also, it's not detectable, just like this one. But when we get the DNA contamination in the whole systems, it means it will show signals just like this one. So if you get a result, NTC results just like this one, it means you get a very high pollution about the genomic DNAs. So in this case, the whole experiment is failure. So you have to extract the RNA from your materials. So we have a box here show the range of the standard data from qPCR. So we have to focus on the TM value, CT value, regression coefficient value, slope efficiency, NTC, and NRC. So there's a range here. So we can say that all the factors is in this range shows this result is very reliable. Okay, this part is the Q&A in the qPCR experiment. First is the CT value is larger than 38. So it means in these signals, we always think the experiment is a failure. So maybe there's three reasons for the phenomena. First one is the application efficiency is too low and the reaction conditions are not optimized. The solution for these reasons is one, redesign the primers or probes, and also the other one is to replace the reagent with the higher sensitivities. And then is to reduce the annealing temperature by using the 3 steps reaction. So maybe annealing temperature can be 30 to 35 or 60 centigrade. And the other reason may be the degradation of the reaction components or low amount of the templates. So maybe you can set the positive control to make sure the whole system works well, and then check the quality of the RNAs, improve the reverse transcription efficiency, and and maybe you can use the one-step qRT-PCR, can be used for a very low amount of the template RNA. The third one is the length of the product is too long. We have thought that maybe the length of the products can be used for 100 to 400. But in this case, maybe you have to redesign your primers and reduce the application products range to 100 to 200 pp. The second phenomenon here is no signals. So there's no CT values. So the whole experiment is failure. In this region, maybe the cycles is less, so you can add more cycles about the whole experiment. Usually, the cycles will run 40 cycles here. But if you detect the virus RNA, virus DNA, the amount of the nucleus is very small, very low. So in this case, you can up to the cycles to from 40 to 50 to detect the whole experiment can be also worked very appropriately. And the other one is the raw signals accuracy settings. The three-step method suggesting extending the acquisiting at the 72 centigrade. And maybe the primers or probes failure. So set the possible control to make sure the primers and probes can be worked. And also the proper annealing temperature is too low, so maybe increase the annealing temperature here. And also the third one is application products can be detected by the gel electrophysics. So if you do the gel electrophysics, you can see very precisely there's whether or not there's application experiment work. The melting curve. This one melting curve is very good result. And this one shows it seems to be very good result here. But you see here, it's a blue lines here. This blue line shows NRC. So it means the no RT control shows the same signal about the experiment. So this one is not as good as well. This one is a small M shape signals, and this one is a very topic M signals. This M signals shows the multiple peaks here. It means there's a non-specific applications during the whole systems. It means the data is not reliable. So we can see this box here. The melting curve is not a single peak. So the first one we have to consider is the primers. So you have to redesign the primers and just use the regular PCR system to detect these primers. Use the gel electrophysics to see the product is one band or two band or smear band. So you can pay attention to these primers with the very normal PCR system. And the other one is the GDA contamination. So optimize the method of the extract RNA and set the NRC control to detect the GDA contamination. And the third one is maybe the gene expression is too low to detect the multiple melting curve. It's not a very single peak here. So it will have to increase the efficiency of the RT system and maybe increase the amount of the templates. One-step qRT-PCR can be considered. And the whole system maybe is failing. So you have to change the reagent. Maybe the reagent is out of the expired dates here. The standard curve. We have to discuss this earlier. Standard curve should be shows a rhythmic sequence here. But in this case, it shows very chaos signals, and also this one. Without these signals here, these four signals shows very good result about the arithmetic sequence. But this one maybe is inhibitors shows is that increased these signals. So it shows very failure result. In this case, we have to deal with the inhibitors. But what is the inhibitor? We have to discuss the PCR, RT-PCR inhibitors as a polysaccharide, organic solvent, and the protein scale as well. So when we detect the quality of the RNA with the nanodrop, these ratios can detect the impurities as an inhibitor for us. In this case, we can do these experiments to avoid the inhibitors. Inhibitors shows its effect just based on the concentration. So in this case, we do the 10 times dilution. So it will release the whole concentration about the inhibitors and shows the good work about the experiment. So in this case, we can do the RNA standard curve, choose the right times for the dilution. And then you can use this dilution to do the whole experiment to avoid the inhibitors in vector. qPCR applications, the final part here. The first one is to detect the gene expression level to diagnose the COVID-19 by N gene or OF1 gene. This one is our product here, SARS-CoV-2 nucleus X. nucleus acid detection kit with the multiple real-time PCR kit. So we detect the 3 genes. This one, RSP, is human gene, so it's an internal gene for the systems. And this N gene and the ORF1A gene is from the COVID-19 virus. So in this case, we do the 3 times multiple real-time PCR to detect the virus. So it's very important applications during the diagnosis systems. The other one is the high resolution melting curve. We call the RHM. RHM is a very specific experiment can detect SMP systems. Not all the machines can do the HRM. It shows that the machine is very precisely control the temperature and it can do the high HRM system. And the other one is the library quantification of the NGS systems. Usually, when we do the NGS lab prep, we can use the qubit to detect the concentration of the systems, but always we have to make sure the concentration, so we can use the qPCR to do the library quantifications. Okay, today's speech is about this one, so I hope these information will help you in your future experiment. Thank you. Okay, there's some questions here. Okay, there's questions. First one, usually I got 1 band of the gel electrophysis of my RNA human cells. So in this case, it means your RNA is degraded. Maybe this band is just a 5.8 band here, just like this one. Very, very large, bright band here. The RNA, when the RNA gets degradation, maybe the 28S RNA, 18S RNA will go very vanished. So you can just see the 5.8 sRNA here. So in this case, you have to extract your human cells with the liquid nitrogen or very good reagent for the extractions. The other one, in your experiment, to isolate the high quality RNA from the formalin fixed. I just answered these questions. Okay, the next one. Then it is suggested to combine both oligo-DT and run primer in RT reaction. Yes, it is suggested to do so. But if you just want to detect mRNA, so it's very precise experiment to detect the mRNA's expression level. You just use the oligo-DT. For example, when we do the influenza virus detection, there's 3 different RNAs. We call the vRNA, cRNA, and mRNA. So in this case, when we detect the mRNA from the influenza virus, we only can use the oligo-DT to detect them. But in normal area, you just want to detect the expressions from the human tissues, from the human cells. Always you can use the oligo-DT and run primer together. But pay attention here, you can use the ratio between oligo-DT and run primer 1 to 1, also can be worked. But we recommend that amount of that is maybe the ratio between oligo-DT to random primer is 10 to 1. It will be very good result here. Next question, why the integrity of my RNA is negative when check nanogel? Okay, through the nanogel, we cannot detect the integrity of the RNA. We have to focus on this part. The nanogel only can detect the impurity and the concentration about the RNA.
电脑的
不是，这在这儿，在这儿。
哦，这个
看一下那个摄像头。
对
啊，看摄像头。
对。
We have to pay attention about this one. Nanogel only can detect the concentration and the impurity about the RNA. It cannot detect the integrity about the RNAs. So if you want to detect the integrity about the RNA, you have to use the agarose gel electrophysics to detect the integrity of the RNA. So this one is the very good quality of the RNA. And also you can use this Angeles machine to detect the integrity of the RNA. So you have to use different methods to detect the different characters of the RNAs. The next question, what's the difference between TechMap Pro method and the hybridized probe method? Okay, here, TechMap probe method. The basic difference between the TechMap probe method and the hybridized probe method is the length of the probe. Usually the TechMap probe here just maybe 20 to 30, maybe little less than this length about the probe. And the hybridized probe usually give nearly to 60 to 70 BP about the whole probe. So in this case, the length between the difference about the TechMap probe and hybridized probe shows the sensitivity about the site mutation about the whole system. So it means when we do the SSD, also you can use the TechMap probe, but you can use the hybridized probe. Can get a very good result about the site mutation detections. The next question, for the multiple qPCR, it is possible to differentiate different samples based on the melting curve. Yes, you can use the melting curve to differentiate the different samples, but you have to use the HRM, this one. The normal melting curve cannot separate the different samples, but the high resolution melting curve can separate them. In this case, the machine shows very precise temperature control. So in this case, the temperature control can control at the 1.0 centigrade at 1 second. In this case, it can show very significant, very small difference between the samples. So we can use the HRM to detect, to differentiate the different samples. The normal one cannot do the job. Okay, the next one. In certain disease conditions, GLPDH and beta-actin levels change, therefore not suitable to be used as a reference gene. In your experience, have you used reference gene other than the 3 genes that you listed just now, and it is reference gene specific, species specific? First of all, the common genes here, the reference gene is these 3, but we never say there's only 3 reference genes here. Usually in the published data, the published articles, maybe there's 70 to 20 reference genes can be used. Beta-actin, beta-tubulin, beta-actin, GLPDH, subwing 1, 18S RNA. So many references can be used. So if you detect the GLPDH detecting can be changed during your experiment, it means your experiment will interference the internal reference genes expression. So you have to choose another one. But you have to choose the precise reference gene based your reference. So based your reference to choose the precise genes. The other one is this reference gene species specific. Yes. Here I recommend the GLPDH detecting and the 18S RNA is mammal species specific. So if you detect a human, a mouse, rat, pig, you can use these genes. But also you have to choose the species sequence. For example, GLPH, you can choose the human GLPH genes to detect the human tissues. But if you detect the mouse tissues, you have to choose the mouse GLPH genes. So it is the species specific. So you have to choose based on your materials. Okay. The factor influence the RNA purity. How to troubleshoot in tissues of the RNA purity. RNA purity is always based on the reagent extraction the RNA. Here we provide a lot of different kinds of reagent to extract RNA. But why? It's based on the different kind of materials, such as this one. Easy pure RNA is very basic one for the normal materials. But if you have a very plant tissue, just like this one, you have used the plant RNA kits or transplant because the whole tissues maybe have polysaccharide or polyphenols. In this case, the very basic one is pure RNA kit do not do his job. So we have to choose the right reagent to extract RNA from the samples. Next one, do I need to include NRC in qPCR? If I already do QC of the extraction RNA using gel electrophysics, yes, you have to do the NRC and NTC in qPCR systems. When we do the QC for extraction RNA using gel electrophysics, it's just that we have to make sure the RNA is good enough for the qPCR. But the NTC and the NRC is to detect the qPCR system is not reliable. These are two different kind of QC systems. The next question, why do we need to use new products combined with fly and Artemis when the EasyCrypt is a higher temperature tolerance? Okay. The EasyCrypt RT is just 42 centigrade. But in this case, we have very basic one here. It's a 42 centigrade. Union one is 65 centigrade. But the whole reaction time is 15 minutes. But the fly Artemis can react just 5 minutes. So we put the two enzymes together, create a new enzymes. It shows both activities about these coenzymes. So it can react at 65 centigrade and only reaction in 5 minutes. So it's a very good product. So I think you can try this new one to dealing with your products. Okay, the reagents can extract both RNA and DNA from the same sample. Are those RNAs good enough quality for qPCR? Yes, it can good enough for qPCR. For example, our products here, transplant and translate. These three reagents can extract the DNA, RNA at the same time. But you have to choose different parts about the whole systems. So you can choose DNA and RNA separately. And the result is also good enough for the qPCR system. And also, in this case, we can extract the protein as well. So in this case, the protein can be extracted from the system, can do the Western blot to detect the protein expression levels. Comparing hepatitis probe and HRM, which is one is better for SMP analyze. Usually the SMP analyze can be used for hepatitis probe and Techiman probe. It's very easy to operate, and there is not to choose the different kind of machines. But the HRM is based on the machine one, so the machine can adjust the temperature very precisely, can do the HRM. So in this case, I can recommend the Techiman probe and the hybridized probe to detect the SMP. It's very quick and it's very easy to operate. It is determination of the melting point is need to be used Techiman probe. No, the melting point is only be used in Sapling 1 systems. The TechMan probe will be destroyed during the PCR systems, so it's no need to do the melting curve here. When we do the melting curve, it will denature the double strand of the products, and it will release the Sapling 1 systems into the systems. So the signal will be fallen when the temperature is go higher and higher. So there's a melting curve exists. But in this case, when we do the PCR systems, the polymerase, the exonucleus about the polymerase will destroy the TechMan system, just to release the signals here. So in this case, there's no way to do the melting curve during the systems. The melting curve just for Sapling 1 system. The next question is acceptable if you use the common reference gene to measure the expression of the small RNA. No, small RNA has a specific reference genes. For example, when we do the microRNA expression level, I think U6 gene is a specific reference gene for the microRNAs. So if you do small RNA, you have to choose specific reference genes, not to use the JPDH or beta-actins. The next one, transcript one-step DNA with GDNA removal and cDNA synthesis mix. In this case, can I use my NRC? Of course, you can use your NRC. In this kit, we also provide another product combined with persistence. We can provide the NRC system during the experiment, so you can also run the NRC in your experiment. The next one, how long can we keep the sample materials in -80 before we track RNA? First of all, we have to make sure the sample materials is treated by liquid nitrogen, and then we transfer these materials into the -80. We can keep nearly half of a year. Will be okay. But if without the liquid nitrogen, you just put your samples into the -80, maybe cause the RNA stabilization. So it's very important about the liquid nitrogen use. Okay, I think I answered all the questions. If you have more questions about the qPCR system, you can put on your screen. I think we can see that.
Hi everyone. Is there any other questions that you would like to ask Dr. Zhao? You can actually put your question down in the chat box, or you can actually turn on your mic and ask Dr. Zhao.
Okay. What is the difference between reference gene and the housekeeping gene? We can say it's nearly the same thing about the housekeeping gene and the reference gene. Housekeeping gene we refer as a JPDH Sapling 1 as a very stable genes expression level, shows very stable. So we call housekeeping. So we use housekeeping as a reference gene. So nearly it's a similar. PCR can actually detect variable or non-variable cells.
活的还是死细胞？
PCR
推荐的
直接可以送。PCR and qPCR just based on the nucleus, so it's based on the DNA and the cDNA. It's not used for detect the variable and the non-variable cells. So it's a two total different experiment. You can use UDG CCK system to detect detect variable and non-variable cells. Okay. Is there a way to check the quality of the cDNA? Unfortunately, there's no way to check the quality of the cDNA. I've heard some clients use the nanodrop to detect the cDNA, and also it can get the signals about the cDNA from the nanodrop. So it will ask us about that. I use the nanodrop, I get the signals, so I can check the quality of the cDNA. But unfortunately, it's a wrong signals. When we use nanodrop to detect the cDNA, the signal is not from the cDNA, it's from the dNTP. So there's no way to detect the quality about the cDNAs. We only use the PCR or qPCR to determine the quality of the cDNA. We prepare a month before in the qPCR experiment. How fresh cDNA sample required for qPCR? Usually, we will request the cDNA as a very fresh one. So in our lab, we always do the RT in the morning, and in the afternoon we do the qPCR. We always kept the cDNA in 4 centigrade just for 1 or 2 days, no more than 1 week. And do not freeze the cDNA in -20 or -80. It will degrade the cDNAs. The RNA stabilization liquid can you mention? Do we use it to treat our sample if we have already treated with liquid nitrogen before freezing? If you use the liquid nitrogen to treat your samples, there's no need to use the iron photo again. Iron photo just used for there's no liquid nitrogen exists. You can freeze the liquids together with your samples. It can be freeze together. Can qPCR be used to detect quantitatively chromosome breaks with specific genes? The breaks occur at the multiple sites with the same gene. In this case, maybe qPCR cannot do the system. We can use chip experiment, or we can use the ATAC sequencing to detect the chromosome breaks with the experiment. So qPCR cannot do the job, but maybe the chip or ATAC sequencing can do the job. Okay. 没有这个哦，又有还是那个，这个should the internal control be included in multiple qPCR? Can reference gene be internal controls? Yes, the reference gene can be internal controls. When we do the qPCR system, if you do the quantitatively qPCR, just like a TechMan system, yeah, maybe there's no need to use the internal controls. But when you use the subgroup-1 as a system to detect the relatively gene expression, the internal control should be included during the whole experiment.
Dr. Zhao, there is actually a participant who unable to hear your answer clearly. So he or she actually asked you to repeat the question again. He or she, the question was whether PCR can actually detect viable or non-viable cells or not.
PCR and qPCR cannot detect viable or non-viable cells. PCR or qPCR just detect RNA and DNA. So it not cares about whether the DNA or RNA from the viable cells or non-viable cells. You can use the CCK or EBU or life and death kit to detect the variable or non-variable cells.
Okay, I think there isn't any more questions from the chat box, but of course, if you have any other question after the webinar, you can contact us, Nanogen Solutions, and we'll try our best to answer your questions. And if there is any questions that we are not sure, we can actually refer Dr. Zhao after the webinar.